Yes. All right. Oh yeah, I need to pull that up. Okay. Okay. All right, okay. Well, welcome to class, everybody. I want to introduce our guest speaker. This is Quinn Bate, and I can give him hugs, and I can call him handsome because he's my nephew. So this is my nephew, who is a medical lab scientist, and he was one of my old students, like forever ago. He sat right there where Remy's at. How weird would it be to be, take your aunt's class? But he was here at Weber High. What did he do? He did football, got some football people. He did choir, was in like chamber choir. Do we have any choir people in here?
Chamber and acapella.
Chamber acapella. He did like the musicals. Artist, like comic artist. Anyway, he was really well-rounded. And that is something that I strongly suggest that you guys do. You want to become really well-rounded. You're just not so focused on, one thing, that you participate in a lot of things, okay? But he's a medical lab scientist, drove up from Harriman to come visit with us today. That's an hour and 10-minute drive. So we appreciate him coming to talk to us, and he'll tell you guys all about what it takes to be a medical lab scientist. So let's thank him in advance for coming. So Quinn Bate, boom.
Excellent. Okay guys, so I've done this a lot. So this is my first rodeo, and so I've done a lot of studies. I'm a scientist, right? So I found the best way for us to get this class started is for us to get to know each other. And clinically, the best way for us to get to know each other is for all of us to come up here and swap some fluids. So I need you up out of your chairs, come over here, and let's swap some fluids with each other.
Swap fluids? You want them to kiss each other?
Come, come, come. Okay, I'm gonna have you guys grab a vial. And anyone that wants to come to the center can come to the center and grab, I'll scooch over. I'm just gonna try and pass as many as I can to help with congestion.
Show them how you swap fluids.
Absolutely.
When?
Okay, so let me show you, demonstrate the appropriate way to swap fluids. Will you swap fluids with me? And that's how it works. It's always best to ask for permission first, so thank you. Okay, go ahead, and I want each of you to go ahead and swap fluids with each other.
How many people should they swap fluids with?
Yeah, let's go ahead and grab like
Two people? Okay, 2 or 3 people swapping fluids.
This is excellent.
2 or 3 people swapping fluids.
Can I swap fluids with you?
Go down, you want to swap fluids? I just support
Can I swap fluids with you? Okay, so I want you guys to go ahead and start finding your seats and everything. Isn't swapping fluids so much fun? It's so much fun. Oh, no, go ahead and hang on to them. We're going to use them. Okay, so this is super fun, right? A little example test and stuff like that. So you guys are super smart, right? So I think by now you guys can guess what I've done, okay? What I've done is I've contended you, right? So we're going to take some of this indicator. I'm going to put a drop in there, and if you stay yellow or green, you're okay. But if you turn red, I got you, okay? So we're going to go ahead and do that. I'm going to set that.
So put a drop.
Just one drop in your tube.
One drop and stir it around and see what color it changes.
Okay, so go ahead. Yeah, it just takes a little bit, and then give it a little caffeine shake or whatever.
If it stayed green, you're safe.
Hey, Chris, maybe you didn't get that. Maybe you were drowning.
Well, I was
Oh,
thinking, that's what I did.
Good.
And so you got it. Okay, so after you get your drop in there, give it a little caffeine shake like you've had too much Mountain Dew.
Survived.
Okay, so everyone that has a green tube, I want you to stand up, green or yellow.
Hold up, we got two
Oh,
more.
two more.
Two more. Oh,
Bro,
gotcha.
dang, bro.
Who else?
Gotta use protection.
Yeah. Oh, safe.
Okay, excellent. Okay, so all you guys that are green, go ahead and sit down. Is anyone yellow? Yellow color? Okay.
That's why you use protection.
Okay. So the idea here is everyone that's red, you were fully exposed and contaminated, right? If you were yellow, you were probably exposed, but you didn't get a high dose. And anyone that was green, you're totally clear, right? You weren't got with anything. So here's the question. I'm gonna go ahead and pass this around. Go ahead and just stick your tubes back in there, and then we'll get it around and pick it up. So let's talk about body fluids, okay? So I am a medical laboratory scientist, okay? And I won't try and get into too much of the detail, because I want to give you guys a chance to write it. But let's talk about this clear fluid, okay? What kind of body fluid? This is obvious water, and I use vinegar, right? So what kind of body fluid could this be? Any ideas from the human body? Clear, colorless, what could it be? Boom.
Like your spit?
Saliva, excellent. We'll come back to that. Any other body fluids we can think of? It's just so beautiful. Maybe tears, right? Or sweat, right? Clear, colorless. Another thing would be your cerebrospinal fluid. It's clear and colorless just like this. In fact, your cerebrospinal fluid is sweet. It's made up of 80% glucose because you guys' brains are working so hard. It needs a lot of energy to be able to do what it's doing, okay? So me as a medical laboratory scientist, when I tell people that I work at a hospital, they're like, Quinn, are you a doctor or a nurse? I'm like, no, I work in a laboratory. So I do tests. And so a laboratory scientist will take body fluids and will test them for the detection, diagnosis, and treatment of disease. Because we want to know the direction that we're going on. We want to know what you have and how we can correct for it, okay? So if this was saliva, let's go back to this side. What kind of thing could we transmit if we shared a drink together with your friend? Any mono. Epstein-Barr virus, also known as, there's a common name for it. The kissing disease, okay? So this is an example of how that would work. So I basically do how people, what they got, where it got, especially when I do flu stuff, we send it to the States so they can track them. So I got mono in college, but that's a different story. I actually gave it to my wife. I guess you guys can understand how that happened. And so what I do as a medical scientist is I work in a hospital setting and clinics right now. You can work in research facilities or for universities. I did that when I was very first starting up. And there's even some commercial industries that utilize our services. It's actually branching out because of some new regulations and stuff like that we can do. Okay? So all these titles are different titles for the same field. They're just different levels. Okay. Are we ready to let me move on? Okay, I'll get it in a minute. So, for example, have any of you seen a TV show called House? Okay, it's this old show, and there's a doctor, and he's like, run the test, and the doctors come in and run all these tests. I can count on my hands how many doctors I've seen in my laboratories running tests. There are teams of fleets of people that actually do the testing. So any time that something comes from the human body that needs testing, whether it's blood, cerebrospinal fluid for a spinal tap, or let's say that your knee's swollen up and you need your synovial fluid checked, stool samples, urines, all that stuff comes down to the lab. And we process it and I analyze it as a scientist, okay? Are we good?
Wait, sorry.
No, you're good. You're good. I always hate it when it's like, it's like a test. I write super fast. A medical laboratory scientist is the 4-year. Medical technologist was the old term for it, and a medical laboratory technician is a 2-year. And we'll get to that a little bit later on. Okay. So this is what I do. I take those samples and I analyze them. And what we're doing is we're looking for detecting the disease. So let's say, for example, I'm in the ER, okay, I get a sample from the ER, there's a patient that's there who we think's had a heart attack. There's a specific enzyme called troponin that I can test for and see what's going on. So if their troponin level is elevated, that enzyme is so specific to the heart, I know it's heart trauma, okay? So then what I can do is maybe an hour later or 2 hours later, we might draw blood again from that patient and check it again. Is that heart marker going up or is it going down? Did they recently have a heart attack or are they currently having a heart attack that's getting worse? Or did they recently have a heart attack or a while ago and now it's coming down? All the trauma has happened, right? So we're looking at the direction. Or let's say like someone's overdosed on fentanyl, right? Scary, scary stuff, okay? So the doctor might need to know that they're on fentanyl to give them Narcan. I mean, so that we can look at the treatment and the direction of what we're doing for treatments and stuff like that. Now in the laboratory, it's a big place. So when I graduated, I first worked for the laboratory up at the university, and then from there I went to the county, worked for a county laboratory for the state, doing drug screens for criminal behavior. And then I got hired before I even got out of school. I was offered a job because I was interviewing. That was pretty awesome to have that happen. There's lots of departments. I worked at Central Laboratory in Murray, Utah, and it's a multi-level, multi-floor, ginormous lab. The entire building is nothing but a laboratory. They recently expanded it. It's ginormous now. And there's different departments. So when I worked there, I would go in the I worked grades then. So I'd go in and I'd look at the schedule and I'd be like, oh, today I'm in urinalysis, or tomorrow I'm going to be in hematology, or I'm doing body fluids today, or maybe I'm doing liquid chromatography, mass spec, and doing drug screens. So I go to that department and I work there for like a 12-hour day, okay? Or a 10-hour day normally is what it was, because I was working 7 on, 7 on. So there's all these different like departments within there. So this is the thing. So sometimes like a nurse will call and say, hey Quinn, I need to know the troponin level. I'm like, bruh, I'm in like urinalysis. Like, I'm gonna have to transfer you to that department. I'm not there today. Because I would run 4,000 samples a shift at the Central Laboratory, especially for chemistry. We were running so many samples every time we were going through there, okay? How many of you guys know exactly what you want to be when you grow up? Okay, how many of those things are still in medicine? Okay, maybe. So let's get this one.
An anesthesiologist.
Anesthesiologist.
Dermatology.
Dermatologist. Maybe
Physical therapist.
physical therapy. Excellent. And then
ICU nurse.
ICU nurse. So when I was sitting right there, I knew I was going to be a comic book illustrator. The reason why I got into the sciences is because this really smart guy, Leonardo da Vinci, studied cadavers and anatomy to help him draw the human body better. I'm like, I'm doing that, yeah. So I got to this class. I'm a very average student, okay? Mrs. Radle's class was challenging, okay? But I applied myself. I think I scraped out like a B+ in the class because I didn't understand some of the techniques that I should apply. And so it was pretty awesome. So I leveraged that and it helped me. If you're going on into those other fields, this is a great foundation, okay? It'll help you. Okay, so let's say that if this room was the laboratory, you come inside and let's say this would be chemistry, that'd be hematology, that'd be urinalysis. I feel like you guys are getting a little glazed over, so let's have a field trip. Let's go over to urinalysis. Come on. Okay, come in. You can get close, I don't bite hard. Okay? Let me know when I'm in frame.
We're good.
Okay, awesome. So this is quite literally how it is. I'd have a colleague maybe working in chemistry, someone else working in hematology, and I would be here in urinalysis. So if we think about it, so I've got some fecal tests that I run at the hospital here, and then here's some urine stuff. Now I want to ask a quick question. Do you guys know what urine is? Any ideas? Because we understand what fecal matter is, right? We eat our food, we drink our water and stuff like that. Our bodies absorb nutrition out of it, right? Any waste products we eliminate, right? And if there's something abnormal with that process, or there's something going on with the lining of our GI tract, we'll catch that in these tests. But what is urine? So if you think of those nutritions that get absorbed, that liquid that gets absorbed into the bloodstream, okay? It gets ferried to the different tissues and feeds the tissues. Well then what happens is that blood then goes to your kidneys and your blood gets filtered, okay? And so really what urine is just the excess water, okay? Your body wants to balance the pH inside of itself. And then there's some soluble waste products like urea that causes it to go yellow or like orange, or maybe extra multivitamins that are water soluble that we eliminate in our urine, okay? Now, you guys haven't done the test yet, but should our blood be sterile? Right? We shouldn't have bacteria in our blood. So therefore, our urine, too, should be sterile, in fact, right? If it's just a filtrated portion of the blood, and mainly just water. So technically, this urine is more sterile than the tap water you drink, okay? Now, people will say like, oh, I've seen Bear Grylls, and he's drinking his own urine and stuff like that. It's good. You could probably do that a couple of times, but it's a concentration, right? Eventually, your body's going to concentrate the urine, and it's not going to do you any good. But pretty neat stuff. And if you think about it, what we can do, you guys will end up doing this later on in your test, you'll be able to do, this is a macroscopic. So these are chemical tests that we can do to perform to look for things inside the urine to see if there's something in there that shouldn't be there, okay? And it's really awesome because we can do this test, find a lot about our bodies without having to jab it with a giant needle, right? And that's pretty awesome, okay? So you guys are going to do this. So let's kind of go down this chemical analysis real quick. So we got like leukocyte esterase. Leuko meaning white, so white blood cells. If you're healthy, should you have white blood cells in your urine? No, so we're testing for an infection, right? Or like protein. You can look at these two samples, they're very different. Do we have any athletes in here? Who's my athletes? Okay, what do you do?
I play volleyball and row.
Volleyball, row? Wrestle. We'll come back to you.
Basketball.
Basketball. Other ones?
Golf and soccer.
Golf and soccer.
Baseball.
Baseball. Soccer.
Football, basketball.
Football and basketball.
Mountain biking.
Michael Miking. Okay, so my endurance runners and stuff like that. I don't know if you've ever if you've ever worked out really, really hard and then you've gone pee and you get those nice frothy bubbles, right? What's happening in that is that's proteins that are getting through. When you work out extraneously or you run or you wrestle or whatever happens, you're putting so much pressure on the kidney and on the filter portion of your body that it's pushing some of those small proteins through, causing your urine to be foamy. Okay. Now, a little bit of protein is okay, right? Because we just worked out, we ran a marathon, we had a match or whatever. So that's normal. But lots of protein could show that net and that filter is breaking down. So that's really good to know. Next thing maybe is like blood. Okay, should blood be in your urine if we're filtering just liquid components? No, right? If you have blood in your urine, this is not good. Go see a doctor. Women, if you're on your menstrual cycle, there could be some blood in your urine, right? Contamination. Something else that we might find in a urine is, has anyone heard of what, or know what it's called when you have glucose or sugar in your urine? There's a disease. Your body's not using sugar. Diabetes, okay? So diabetes. So there's two types of diabetes, right? Does anyone know anyone with the first type? Okay. Is it like a relative or something? Do you know anything about type 1?
Not really, no.
Okay. A friend? Okay. So type 1 is diabetes insipidus, okay? And what this is there's glucose in their blood, and insulin needs to bind, like the key, to open the door to allow glucose to come into the tissues to be used. Type 1, they don't have insulin. They're insulin dependent. Their body's not producing the insulin or enough insulin. So they need injections, right? So once they get the key, door opens, glucose comes in, the body can utilize that glucose. That's awesome, right? Type 2 is when, due to lifestyle, where the adipose and the fats stretch out the receptor. Now you have the key, right? You have this insulin to bind. It just can't bind because the receptor's messed up, okay? And the glucose can't be brought into the tissues. And your body starts eating the next best thing. So if you can't eat the sugars, what does it go for? Not fats. Fats are hard to break down. It goes after muscle. I can actually smell the ketones on someone who's in ketoacidosis. Because the body wants to survive, no? It doesn't want to die. So that's something that we'll do. We'll also test for that. So we can tell a lot about what's going on in the body. Now back in the old days, so we have these chemical tests that we can dip and we can check, right? But back in the old days, diabetes mellitus, meaning syrup or sweet, what they would do is they didn't have the chemical tests. So back in the old days, what they would do is they'd actually test and taste. Apple juice. To see if it was sweet.
It
is not pea, it's apple juice.
It's apple juice.
I was just like, hold up.
Yeah.
Wait a second.
So they would be able to diagnose what was going on. Alcoholics, I've smelled ketones, but alcoholics, I've smelled alcohol on urines before. I could smell even drugs and stuff like that in some quantities. So really kind of interesting thing. So there's lots of things. You'll get to do this later on this year and do some chemical testing. Now, what I might end up doing later on is actually, if there's something abnormal in here, okay, I might need to go ahead and spin it down and actually look underneath the microscope to see what else is there and quantify those types of things, okay? There was one year where I was doing my rotations and there was a really bad urine. It was really bad. You couldn't pour it. It slithered around. There was so much bacteria and white blood cells in there because the white blood cells had fought the infection until they died. So it was stringy, mucousy. It was terrible. I had to cut it with scissors just to get it on the slide. Yeah, it was bad. So this is a swarming Proteus, and it smells terrible. So I did the count, got it done. We couldn't leave it on the scope. We had to take it off the scope and put it in the bucket, and then put the lid on the bucket and take it from the room. It was terrible. Even took the cup out. So pretty cool stuff. Okay, questions, concerns?
If
And-
you have
yeah.
diabetes and the glucose in your urine, will that make your urine thicker?
No. So that's a great question. So glucose is water soluble, and that's why it comes through the filter. There's another silly thing that you can do. If you're on, so if you take a multivitamin, your urine might be that bright yellow color. I mean, it looks like a neon marker. Okay. Or let's say if you're on methotrexate for an infection, or pyrimidine, your urine might be orange or green, right? Or when I was in college, I played a joke on my physiology professor, and I took some methylene blue dye that's water soluble, and it turned my urine blue, like Smurf blue. And anyway, yeah, she didn't think this is funny. Anyway, so cool. Any other questions? Okay, concerns, insults. Awesome. Let's head back up. And for the record, the urine was worse than you think it was. So whatever you're envisioning, it was worse.
So Quinn, when they do drug tests, are they then just doing the urine?
Yeah, so what we'll do with the drug tests, and that's the thing. So methods is what it's based on. So there's a methodology to it. So we have macroscopic and microscopic for our urines. And then we also have chemical analysis. So I can run a color metric, so the colors will change, and it'll let us know for a specific analyte what the concentration is based on some nerd math that we do with light and colors, okay? And that lets us know what drugs we're on. So we'll run a drug screen. If those drug screens are positive, and I normally joke and say, hey, I'm giving every student in here a random drug screen. Watch who freaks out. Because if we find drugs in there, we'll send it off to that LCMS, mass spec, the liquid chromatography mass spec, and we'll actually break apart the atoms and actually know exactly what's in there. We will know the parent, the metabolite, the daughter of everything. So if you go to the hospital and you are doing drugs, just tell us. We're there to help you, and we're going to find out, but it's going to take some time. and we can't help you unless we know what's going on. I've seen people, because I worked for toxicology and I worked for the hospital, people were putting water in there and like turpentine and stuff like that. Come on, I mean, just be adults about it and let the physician know what's going on, okay, in your life. So, that's a great question. And in that sample, each one of those, the samples destroyed, some samples we actually retained them. So a normal day for me, when I moved over from Central Lab to Altoview Hospital, I became a chemistry lead. So I did all the orders, all the orders for the supplies. I was on the chemistry work group, so I made policy for all of Intermountain for the chemistry department, and I would pull the reports. So my normal day would start like at 4:12, 4:15 in the morning. I would get up in the morning, I'd hop on my motorcycle and drive up so I could be at the hospital at 5 a.m. Sometimes I'd have to check the schedule to see if I was actually in chemistry, because I am a generalist. I worked all the different departments in the laboratory. And so let's say I check it, I'm in chemistry that day, so I would do my maintenance, okay? So this would be doing my cleaning, I would do any of my documentations in the laboratory, and under regulations, if you don't document it, it didn't happen. I would go ahead and do any adjustments to my machines to make sure they're working properly, and restocking my reagents, consumables, and checking our water. Even the water that we use has to be a chemical grade water. We can't just use tap water, because the metals that leach out into the water will actually cause the results to be bad. So what we do in that is we do what is called a calibration. Okay? And quality control. So if you think about it, the big machines are made up of probes, pistons, pumps, lamps, lasers, or even like regulated temperature baths and stuff like that. So there's our analyzer, okay, on one side. The other side would be our reagent, and that's a specific reagent. I would run like 156 liquid assays, plus my 25 external annual assays, and then a plethora of like PCR, which is where we amplify genetic material for like COVIDs, okay? So what we would do is I take that reagent. How many of you guys like chocolate chip cookies? Okay, is it chocolate chip or chocolate chunk? Do we like chocolate chip cookies or chocolate chunk cookies? Chip? Okay, you guys like ratios. Okay, I love it. So if you think about the reagent, when you make up a recipe, let's say we get our bowl, and we put our flour in, our eggs, our sugar, everything, we mix it up, and we, 1 cup, 2 cups, 2 cups of chocolate chips, right? We mix it all up and we make a batch of cookies. And we're eating our cookies, we're like, oh, these are so good. But then we're like, hey, let's make another batch. So we follow the exact same recipe, and we put our ingredients in, and we bake those cookies. Are they going to be exactly the same? No, there's going to be a slight amount of variance, right? Well, that's what calibration does. So if we change something with our machine, we have to reset up that relationship between the analyzer, all those things, and the lot that we have. And sometimes those changes can be big. Then what we do is, after we establish that range, we want to qualify it. I want to prove that I have control over how my analyzer is working. So we'll run a sample at the low end, in the middle, and at the high end to prove that we have control over the machine. It's giving us exactly what we want within a range, plus or minus some standard deviation. Nerd that, okay? So once we've proven that, now I have spent an entire day fighting with a machine that had problems. Because if I change out a lamp and yank it out, put a new one in there, well, that old lamp may have degraded over time, right? And the new lamp might be giving me way more light. And so I have to adjust for that. So I've fought with machines all day long, and then I have to do this regardless if I see one patient or I see 1,000 or 4,000 patients, right? This has to be done based on regulation. So let's say that we have a good day, okay? And my controls are all in and everything's great. So that means I can start testing. So I might start loading. Let me grab my samples. Can't find them. I lost them. Okay, so I grab my samples. We'd spin them down, we'd go ahead and load them on. I'm like, okay, I've got multiple tests on these patients, and load them on. And then maybe there's a nasal swab that comes in. There's this thing no one's ever heard of it called COVID. You ever heard of it? Okay. Anyway, so I'd set up like a COVID test, right, on the PCR. And then I might have like a mono test, right? So I'll go ahead and take a serum, and I set up a timer and run the mono test. And I'd be running back and forth, taking care of all the patients as they come in. I would result them and let the doctors know what's going on. Now, let's say that we have a diabetic and their glucose is too high. It's like 200. And it's saying it's greater than 200. And it's like, I can't count this, there's too much in there. I might have to do a repeat test, so I might have to go in and do a dilution, right? To bring it back down so I can actually measure it and then multiply it by the math. Or let's say that we have a bunch of teenagers who are out having a good time, go eat some hamburgers and some French fries. Their blood gets all of the fats in it and stuff like that, but then they get in a car accident. They come into the ER. How dare they not have a fasting sample? So when I spin down their blood, it's all milky, which would scatter the light. Well, I would have to treat that to clarify it so I could actually get the results to find out what else is going on in their bodies, right? If there's any other trauma. So based on the way or the method that I'm teaching, I'm doing my analysis, I might have to do that. So I'd be setting up manual tests, and maybe there's new machines or new tests that I have to do validations on. Me being a lead or being the coordinator for the chemistry department, at the View Hospital, actually corporate-wide, we're actually changing the method that we're using for our chemistry analyzers corporate-wide. And so we're having a huge change that's going on right now. And then let's say that Tatum or Reza is like, hey Quinn, we gotta go to lunch. So I would do my department and cover their department at the same time and run back and forth, trying to triage everything. Okay? Questions, concerns? Okay. Okay, so within the multiple departments, let's actually do we did the field trip to urinalysis. So this is like urinalysis after we would spin it down, okay? So we had an issue with the urine, right? And so here's a picture of the red blood cells that would be in there. You can see these red blood cells? They're all shriveled up. Okay, you guys are smart and you guys have done a thing. What would cause these red cells to shrivel up like this? Because normally they look like this in the blood, right? In the serum, and it's all thick and has all this stuff. So what could be going on with these red cells? Speak up, you got it.
I don't know if it's,
Yeah, go ahead and try.
I don't know if it's something, but would it be like hypertonic?
Yeah. So what's happening is so the urine is super aqueous, right? Well, inside the red cells, it's got lots of potassium. Well, it wants that gradient to be equal, right? And so actively, those red cells are pumping out the potassium to try and equalize that. Now, the urine, there's so much water in there, they're not going to. So they start creating like this and shriveling up. Now, that lets us know that that takes energy to do that, right? So the blood has been there, and it's been there for a while, right? Same with like, if there's it's called a subarachnoid bleed, we can see what's going on with if there's red cells, because you shouldn't have blood in your brain, right? So if we can see that, it tells us what's a clinical picture of what's going on. Also, these are calcium oxalate crystals. So when I worked at the hospital, at the central laboratory, I worked grave shift, 10-hour shifts, sometimes 12, 14, 16 hours is normally where they cut us off, because you go loopy a little bit, and you're a little bit dangerous. Well, we wear lab coats, we have gloves, and you have to wash out and scrub in, basically. And so we couldn't have water. It was all sterile. Well, not only sterile, but we can consider contamination. So a lot of us didn't get to go out and get water breaks. There was only 4 of us on the grave shift, and so there's about 5 of us that all got kidney stones, and it was terrible. So these kidney stones, your kidney concentrates, right? So if the oxalates don't get bound up in your GI tract and eliminated, they precipitate in your renal pelvis, and then they turn into shards of crystals that you get to pass, and they get to cut their way out as they go. They can get too big, and then you actually have to get sound to break them up. Or they can get so big that you actually have to cut them out. Dr. Gradagraph actually had a kidney stone that was this big spike ball. The renal pelvis had milked it into the shard. And if you licked it, it was like salty, and I'm just joking. But they can get so big and so fierce that they can actually block the kidney from doing what it needs to do. Now, yeast we might find in the urine as well. So women, you have a shorter urethra, that's just biology. and so you're more susceptible to yeast infections. Men, as we age, our ability to fight off infections diminishes over time as we age, so that can be common. So we want to make sure that these are out. And then based on these things, if we're finding bacteria, we can prescribe different antibiotics to do that. Which screen am I on? There we go. Okay, so microbiology, it may be another thing that we do. How many of you guys, what's something that you don't like to eat? I mean,
Broccoli. I don't know.
broccoli. My dad hates broccoli too. Anything else? Tomatoes. I had a best friend in high school. He would throw his tomatoes out of his window of his truck when we saw that. And I was like, I'm not eating that. So I can't stand onions. Onions are kind of like that food that you invite that friend over to the party and they talk over everyone. And then it starts getting late and you're like, it's like 11, 12 o'clock. You're like, Onion, you can go home. And then you get up in the morning, Onion's still on your couch. You're like, Onion, just go home. That's what onion is. So, I like onion. But bacteria is exactly the same way. They're picky eaters. They're fastidious. So we have these different plates with different stuff on them so that people who don't like pickles, you know what I mean, or the bacteria that don't like blood agar won't actually grow, and so on and so forth. Now we want to isolate what's causing the infection, and then we want to identify it. We can use some stains. But mainly what we want to do is a culture and sensitivity. We want to know what's working well, right? We want to see what's doing well. This is why we don't want people just to take antibiotics on their own. Because if we can isolate and do a sensitivity, we can find out where the zone of inhibition is best, okay? So what we will do is we'll set up a Kirby Bauer plate and put drugs on there. We have new methodologies to do this. But you can see that some drugs are effective and some are not. Now, if we take A on the Kirby Bauer plate, it's not going to do us any good, whereas E is working amazing. But look at F. F's a bit weird, right? It's taking care of some of it because the brothers and sisters are going. So some of them are susceptible, but some of them aren't. So if we do this, if we take drugs before we actually can isolate it, we can start developing resistant strains and leave them behind. These superbugs can actually cause us lots of problems. Have any of you heard of like a MRSA, a methicillin-resistant Staph aureus? There's even some new stuff, inapinum, which is a horrible drug that we use to sterilize people. It takes care of like all your normal flora. It's actually really, really bad. So what do we do with people that can't? I had a Pseudomonas that the entire lawn, the entire. So what do we do with these things that we can't treat with drugs? Any ideas? You end up having to cut them out, right? And when you cut out healthy the contaminated tissue in the bereavement process, they actually have to take some of the good tissue with it, right? So they have wound clinics all for this if that happens. So hopefully not. Okay. Cool. Okay, so another department that I might have, it would be like hematology. And I feel like you guys need to stretch your legs. So should we all go together over to hematology? Let's head. Okay, raise of hands. Can I have a volunteer to help me with a process over here in hematology? Who's okay with blood? One so far? Okay. Will you come over here and help me? What size gloves do you wear? Okay, we'll try mediums. What's your name?
Crew Phelps.
Nice to meet you, Crew. Okay, go ahead and put on those gloves. Okay, so hematology department, we have an analyzer. And so this is my blood. I got this drawn by a friend of mine. She drew it yesterday for me. And so what we would do is we take this, you can see it's separated right now. So we go ahead and make sure that blood's nice and mixed up. Because we want the ratios to be, to simulate what's inside of our bloodstream.
What's separated?
So that's a great question. So about, depending on your gender, for males about 40%, 45% is packed red cells that carry oxygen. Then there's it's called a buffy coat, which is white blood cells. It's like 1% that is white cells and platelets, okay, to clot. The rest of it is your plasma or serum, okay? And that's the matrix that's carrying the molecules and the nutrition and everything like that. That's a great question. And then inside that too, there's other things in there that helps clot our blood and things. This is in an EDTA solution, or it's really a powder, EDTA, and it's binding up all the calcium in here. So this won't ever clot. And that's why we use the different things. Like heparin will bind thrombin, and there's different things that we use to actually do that. That's a really good question. So we want to mix it so it resembles our blood. But what we would do is we'd run this on the machine. The machine would do a lot of light, math, and stuff like that, and give me numbers. So it would tell me how many white cells there are, how many red cells there are, what the ratios of those things are, and let us know. Let's say that there's something abnormal about the blood, okay? So what we would need to do then is actually do a microscopic, or do a slide. And that's where, is it Crew? He's gonna help me. So he's gonna do a smear with me. So if you guys can, without blocking the camera, if you wanna come in kind of close, because this is gonna kind of be small. So this is, I call this lovingly, it's a helicopter top, and it's just a chamber that opens up. So we're gonna come closest to me. I'm gonna tip it upside down after it's nice, well mixed. And we're gonna push, I'm gonna let it fill into that little tube, and I'm push down and make a little drop of blood, okay? I'm gonna demonstrate first. So then what I'm gonna do is with my main hand, I'm gonna hold onto this slide, and I'm gonna anchor the bottom slide with the blood on it. I'm gonna rest this on here, and it's just gonna ice skate back and forth, okay? I'm going to drop back, I'm going to touch blood, it's going to flash across, go. And then I'm going to push it forward. Nice and smooth, slow as smooth, smooth as fast, okay? Without letting it teeter or tip. And I want a thumbprint. You can see my thumbprint's a little bit off because of how the blood's flashed across. But you can see that kind of feathered edge on the blood smear. That's where the red blood cells, the white blood cells are actually stacked right next to each other. We don't want them all crammed together like on a subway car. We want them nice and spread out so we can see everything. So Crew, can I have you go ahead and give that a go? So you're gonna, are you right-handed?
Yes.
Okay, so go ahead and put your drop of blood close to you. The smaller the better when you're brand new. Excellent, like a boss. Okay, and what, with your primary hand, I like to hold it so that you can adjust pressure and just rest it on there. And go ahead and decrease your ankle just a little bit. Draw it back, touch your blood, watch it flash across. Now push it forward nice and smooth. Keep going. Okay. Now I will tell you that, okay, so he almost has a reverse thumbprint. Do you see that? This took me a whole semester to get good at, okay? This is kind of a difficult technique. Thank you so much for doing that. So we smeared out the blood. We could actually take these samples and put them in a stainer and stain it, and it would separate all the different cells and cell types. And if that were the case, I would have all of you come here, we'd all make smears and slides together, we'd stain them, and they would be completely inert, and I could give them to you, you could take them home. Your parents would look at them and weep with how amazing their children are and hang them on your fridge, you know what I mean? And all that stuff, but we don't have the stains to do that. Baby blood has fetal hemoglobin, and the extra proteins to like deal with being born is traumatic, and so their blood is super thick, and that fetal hemoglobin helps extract off the white cells from, the oxygen from mom with affinities. So baby blood is super, super thick, so I might have to really decrease the angle to have it push long versus keeping it tight to have it go short. So thank you very much. I'm going to teach you all. I'm going to actually put these in the Sharps pen and the biologics so they can autoclave. Let me show you how the proper way of taking off gloves. You're going to take your glove, you're going to turn it inside out, and it's going to Pac-Man and eat this with glove. And we're going to grab this glove and turn it inside out, and that's how we sterilely take off our gloves. Great. Any questions about this process? Sweet. Thank you so much for helping. We can go back to our seats. This is pretty fun, right? Not too shabby, okay? Now let's actually think about this. So those slides we would make, this is what it would look like. We could stain it, the red cells would look red. The white cells, this is one lineage of white cells. There's more white cells than this, but this is one spectrum of white cells. So if I were to tell you 100 or 200 years ago, then I'm going to take your blood, I'm going to put it on a piece of glass, and I'm going to look at it and tell you if you're going to be okay or not. They would call me a witch, right? Because that's what it sounds like. But that's basically what I do at work, is I take people's blood and I check them out. So here are neutrophils. In a normal healthy adult, about 45% of our white cells are this neutrophil. This is a mature neutrophil, okay? These are eosinophils, and these all come from the same lineage. And this is an eosinophil. Anyone allergic to anything? Boom. What? Mint? Ugh. Amoxicillin, Saki.
Pollen and bees.
Pollen and bees. So these eosinophils are the type of things that deal with allergic reactions. I'm allergic to cats, so I had to stop eating them. I'm just joking. Also in like, what do you call it, parasites. So if you had giardia, a pinworm, or tapeworm, you might see an increase in eosinophils. So based on looking at someone's blood, I can tell about what's going on in their system. Basophils might be if you're allergic to grass or something like that, and you have hives and stuff like that, basophils are a part of that process too, okay? So, pretty neat. Red blood cells, what do? They carry oxygen, right? So if you've ever gotten a bloody nose and you can taste that like metallic-y taste, you know what I mean, in your mouth, it's the hemoglobin, the iron in the hemoglobin that you're actually tasting. These are kind of shaped like donuts or like tubes with like a center in it to provide as much surface area as possible and carry as much oxygen. We're at a high altitude right now, right? So the air is thin, so our bodies actually has extra cofactors built into it to help us with nerd stuff, manipulate the structures to help us utilize the oxygen better. It's pretty amazing, okay? Now, any of you guys, so I graduated in 2001 from here. I think it was the next year, the year after. They came out with a really old movie called Lord of the Rings. Yeah, anyone seen it? A couple, okay. So in the second movie, what happens is there's a battle that happens on the way to a keep called Helm Deep, and all of the adults, or most of the adult soldiers, die. So who do they give weapons to fight in the next battle? No one remembers the movie? So they go and they grab the high school students, so a band, and then they start grabbing the junior high students, and then maybe they grab the elementary kids, and if things are really bad, maybe they start grabbing the toddlers. I mean, to do whatever. The body is very resilient and wants to survive. So this left shift, I can look at and see if the body's bone marrow is reacting in a specific way to see what's going on. Now, if things go haywire in the bone marrow, though, then we get into cancers. And that's the bummer thing. The other thing, too, is I'm looking at your red cells. They carry oxygen, right? So if I see something that's going wrong, there's genetic things, or in this case, if you start clotting inside your body, it's worse than if you start bleeding and you can't clot. Because if you start clotting inside your body, well, those fibrin strands will start slicing up your red cells. And that is no bueno. And so bad things can happen there as well. Okay, we want to keep people alive. Also, if you think about it too, so oxygen, red cells live for about 120 days. And so this is where carbon monoxide is so dangerous, because it has a stronger binding. And as long as you can hold your breath for 120 days, you should be fine. Your body will make new ones. But that's where carbon monoxide is so dangerous. It's because of how it binds to the red cells. And then obviously I'm also the first person who sees like cancer or leukemia. And so I'm the one who's calling the oncologists and the doctors to let them know that we need to do further testing to identify what's going on. Okay. So if I have a really interesting day at school, at work, someone's having a terrible day, if that makes any sense. Okay, so we also do blood bank as a generalist. Blood bank, you will later do your typing and stuff like that, and find out what blood type you are. I'm A positive. Blood is regulated as a drug, and we want to make sure that the right blood goes to a new recipient who matches. And if they don't match, it's actually the quickest way you can kill someone, because your body will see it as foreign and attack it. What's that?
Can you go back to the last slide just real quick? And
Yeah.
write that down.
Let me see.
Thank you.
Yeah. Awesome.
So that's showing what cancer looks like.
Yeah, these two are blasts. This is cancer cells. This is called CML, and this is a chronic lymphocytic leukemia. And they look like soccer balls, chocolate chip cookies. And so, just like you guys are doing your tissue stuff, my brain, I'm taking pictures in my mind. I was an artist when I was doing my stuff. When I took anatomy, this class prepared me for anatomy really, really well. I didn't take list notes. I would draw pictures. That's how I got through physics as well. I would draw pictures because my brain works in pictures. So I know what CLL looks like. Also, because of the osmotic fragility and the cell structure fragility during this leukemia, these little popped cells are called smudge cells. That process of us mechanically sliding out the slide actually will break them. It can actually get so bad that I might have to add into my blood draw albumin to protect the cells so that they aren't destroyed during that process. Now, that protein coat will change the way the stains work, so I might have to have one stain to do my count for my ratios and do a separate stain to look at how they look and the weirdness that's going on with them to identify them. Okay? Now we can send them off for flow and see. Because really this is like take an 11-year-old, a 12-year-old, and a 13-year-old, and stand them all up here and say, I want you guys to tell me which one's the teenager. Right? Kind of difficult, right? Because you can be like, well, she's tall, he's short, I don't know. It looks, there's similarities there. Okay? Great question. Okay, so education. So there's a campus route to people go on campus and actually like talk with the professor. I love that. That worked well for my brain. So when I was on campus, I could go and say, hey, Mrs. Radel, I don't understand this glucose thing. Can you explain it better to me? I mean, where some of you, your brains might work better with working online. If you can read and get through it fast and understand what's going on, then maybe that would be better for you. So there's a progression with medical laboratory sciences. First semester for the program, you can take 2 classes. There's a urinalysis class and a phlebotomy class. Once you do that, you're basically a medical laboratory assistant. You have a certificate for that. You can go and be a phlebotomist or a specimen processor. You can go touch all the plates that we do and all the samples that we set up. They're our front line. Part of it is what they want to do is, I'm a sciencey guy, right? And to spend the kind of money to have me go and do draws and set up all the samples and tissues and wounds and all that stuff doesn't make the hospital as much money as if we have someone else doing it. So then what I could do is you would apply to the program to get into a 2-year associates. And you would do all the things we talked about. Chemistry, you do blood bank, you do hematology and more urinalysis and get into that. After 2 years of education coursework, you could take a test and get qualifications to be a medical laboratory technician, okay? And you could continue to work in different fields and earn more money. Later on, you would have to reapply for the 4-year degree and get back in. Then they go over the exact same stuff that you went through before, but deeper. They cover ethics, also a lot of like managerial stuff, supervisorial stuff, reports, records, and stuff like that too. Also, as you go on with your education, so a 2-year degree, all of these work under a pathologist's license. Now, as a 2-year degree, you might only be able to one specific test, whereas as a bachelor's, actual full-blown medical laboratory scientist, I am considered an advanced medical laboratory scientist, and so I ran a department. Okay, so that gave me abilities to be lead or a consultant or to be a coordinator. Okay, and so it opens opportunities. Those opportunities, we'll get into like monetary a little bit later. I went to Weber State University because they did simulations labs. My brain worked really, really well where I'd learn the work and then go to a lab and apply what I learned, right? Instead of reading about like working on something, I would learn about it and then do it. I'd learn about it and then do it, which worked really, really well for me. Because those simulation labs were like running in a simulated hospital, we would have short hospital rotations. I'd go for 1 week, for each department, and I'd run through those pretty quickly and be done with those rotations. My senior year, I got to do research, which was really awesome, because I got to apply my sciencey brain, and it's kind of being in the jungle with a machete. You think you've thought it all through, and you're absolutely wrong, because I spent 3 years developing the plan, and I kid you not, we ran into so many issues, it was crazy. But I got research funding, multiple thousands of dollars research funding. I believe it was $40,000 or $50,000 research. University of Utah, Brigham Young University, they do things differently. There's one other university that has recently come on that actually does medical laboratory sciences degree. But what they would do is they would do all their prerequisites, they would get into the program, and they would do 2 years straight of nothing but theory. Theory. Reading books and all that stuff. Then they would spend 6 months at the hospital just trying to get all the experience that I got in the last 4 years, if that makes any sense. And for my brain, I went to Weber State because that was way better for my brain, okay? One of the biggest advantages that you have right now here in high school is you need to learn how you learn, really, and establish that. Like I said, I would draw pictures to study, and it worked really, really well for me. Come up with a pattern, okay? So you need prerequisites to apply. Now, when I was sitting in that chair, I was like, I'm gonna be an artist. I only need enough anatomy and physiology to get me into art classes in school. Well, I didn't have my chemistries done. So when I applied for the program, they denied me. So it took me 5 years to get my 4-year degree, because I had to wait 1 semester, which took a whole year to reapply to the program to get in, okay? So you have to do those. Long laboratories, there are lots of laboratories that would go sometimes till 7. When I was doing research, 9, 10 o'clock at night. So. Any questions?
Thank you.
Okay. After the end of that, 4 years, I would take a national registry exam. But during the education part, we would take tests on the subject matter. And we would need to pass with an 80% or better. How many of you want someone who gets D's to do their analysis to see if they're sick or if they have cancer? Nobody, right? So they have us have an 80%, that's the same exact for this course to get the credits that you need. You'll need 80%. If you were to fail that test, and you could have a 79.9, you would be offered a similar type test for retake, okay? If you were to fail that test, they'd give you one more chance on the subject matter to take a test of a similar type. If you were to fail that one, they would excuse you from the program, because they really, truly cannot have people who couldn't do at least B-type work in a hospital setting, okay? Now, I'm the most average student you will find. I got like a B+ in my aunt's class. She was super hard, strict, you know what I mean, because she didn't have any nepotism with me being her nephew. But I figured it out. Had I had a better structure for like studying and knowing when things were due, I would have been fine. I accidentally got on the honor roll and high honor roll at Weber High when I was here, okay? But I didn't know how to apply myself properly. Any of you guys doing concurrent enrollment or PA class, what is it called? AP classes? Okay, all of you guys that are doing that, you're saving yourselves thousands of dollars. On top of that too, the concurrent enrollment means that the efforts that you're putting in now are actually going to credit. The AP exams, you're putting all your eggs in passing that one test, and all of your work is for naught, right? Now both of them have benefits, and just realize that you have an opportunity to save yourself lots of money, and don't be a dum-dum like I was. I took out student loans. It took me about 10 years to pay off my student loans, okay? You can actually get yourself in a situation where you can actually have people pay for your education, and you can just open the doors. Because once again, these doors don't close with the education that you get. Education can only open doors for you, not close them, okay? So I was in my senior year, and I had this big calculation that I had to do for chemistry. It was multiple pages with lots of Excel spreadsheets. And I remember the professor saying, Quinn, you've got to make sure that you present these all just like, you know, when you have your format. Make sure that you express everything as a percentage. But I forgot that I built the percentage into the formula. So when I selected it and said express as a percent, Excel multiplied everything by 100 again. So I was off by a factor of 100. So I got a 0 on the entire project. So I was offered a retake, which was to resubmit it. And I went to my professor and I was like, oh, this is so unfair. Like, I just made a simple mistake. He's like, this is how rural hospitals work, right? So I changed that application, resubmitted it, got 100%, but I only got an average, which is 50, right? So I got 50 on that examination. Luckily, I passed the retake, but between that and organic chemistry too, it cost me graduating with honors. I was kind of bummed about that. So, and like I said, I'm the most average student you'll ever find. So, figure out a way to keep track. What I would actually do in college is I would take a normal sheet of paper and I'd fold it into three sections, and I'd mark out my entire semester, and I'd put out the weeks, and when a professor said, this is when this test or when this was due, I would mark out those days and say, this is the time that it's due. And then if a professor said, oh, we're going to move that test, I would switch it. So when I'd open my binders, I could see exactly what was due. And once I could keep track of stuff, that's when my grades did really, really well, and understanding how to do it. Okay, so the American Society for Clinical Pathology is the comprehensive exam that you take at the very end of everything. I took this test. It's like a 3-hour terrible test that you bleed out your eyes. It gets progressively harder. So if he asks you a hard question, you answer it correctly, it'll ask you a harder question. And then it'll ask you a harder question. And you only have to get 60% because everything in the 4 years, anything they've ever whispered about is fair game, right? And so if you're doing, like, you're sitting there and you're like, oh, I'm failing this test, this is not good, you're actually doing really, really good. If it's getting super easy, you should be nervous. And it doesn't tell you when you hit that 60% pass rate. It's much like the nursing test, the NCLEX. So after you pass, you have your medical laboratory science certification. Then you have to do continuing education and pay money and take tests to continue to certify. Now my hospital and clinics have me doing testing every quarter. I revalidate what I do, then I submit them to a governmental agency to prove that I know what I'm talking about. Okay? And that education is continuing ongoing. Also, the methodologies are changing. What's up?
When do you take this test?
So there's you can take a test like this after your MLT. It's a great question, but most of us take it after our 4 years so that we get the full thing. Because why pay for it twice? Does that make sense? I was offered an opportunity to get my 2-year degree. I wasn't planning on taking it, but I was offered a chance to do it for free, and free is awesome, right? So I went ahead and got that. It also, that NACL accreditation didn't expire, and it didn't require any renewals. So I went ahead and took that test. It was just before I took in my national registry exam. But my arc was a little bit different because I was trying to go into medical, dental, or PA school. But we'll come to that in a second. That's a great question. Also realize that in these fields, as a medical laboratory scientist, I'm a generalist. I like the variety of being able to work in multiple departments. Now I work for clinics. I'm the regional technical consultant for the North Salt Lake region. Okay, so I go from Salt Lake Clinic all the way out to West Valley and down through Taylorsville. Okay, then I'm helping because I was the lead chemist for Altitude Hospital. I'm helping them with their validations up there, and I covered their hospital when needed. I also worked the COVID redeployment, department when that happened, and then I worked at Heber and Riverton and these different departments as new ones came through. So, or what you could do is you could go and specialize and do something that's very, very unique, like a rub, and like a reference laboratory, and be really good at one thing and get your depth of knowledge. There's good and bad to both of those. Me as a generalist, I felt like I could spread out my knowledge base and be more of an asset. I got to dabble in a lot of cool things, but I'm also responsible for keeping up my competencies and all those things. Whereas someone who is specialized only has to focus on what they're focusing on, right? Now, I've been passed up for promotions by people who were specialists, right? Because they didn't look at my generalist application as being valuable. And that irritated me. So, okay. Oh, great question. Let me go back. Okay, so pre-professional. Weber State University has a pre-professional track. So I actually did all of my prerequisites for medical and dental and PA school and genetic counseling alongside with my medical laboratory sciences. I wanted to open the doors as wide as possible so that I could go into dental school, medical school, or PA school. Now, a lot of my friends went on to medical school, but that's one of the reasons why people using medical laboratory sciences as a stepping stone to go into other things, that the career needs more people. Also, what happens is people will leave the field going into laboratory information systems. So if you're computer-wise, and because all of these machines have to talk to each other, you might do that. Or let's say field service engineer. There was a young lady in this class, I actually knew her dad, because he's one of my field service. So those lights, pumps, pistons and stuff like that, I would work on it to a point, but sometimes we need engineers to come out. So they hire engineers to do that. I had another good friend who actually wanted to be a PhD. He wanted to do presentations, research, and university work. Pathology assistants, looking at, that's like a master's program, professor, or working like toxicology specifically, and mass spec, or doing research. So you can see that this field gives you a lot of variety. And if you find something that you don't like, you can make a career switch. I recently felt like my accolades weren't being utilized properly at the hospital, so I switched over to be a technical consultant for the clinics to leverage my skill set. Also, I'm actually going to be working on my master's program, and that's why I've made a little bit of a career change. So, highly recommended if you want to open up your opportunities. So the projected increase over the next couple of years is about 11%, and I bet you these numbers are a little bit lower. Also, I'm the young guy, okay, in the laboratory. The last 2 hires that we hired at Altaview Hospital were from, they were a biology major and a microbiology major.
We only have 5 minutes.
Okay, perfect. And so what that does is, there's a greater need. We also have aging population, me being a young guy. Next 5 to 10 years, we're going to lose the majority of our advanced medical scientists. A lot of my friends have actually started to retire. Now, variables, technology, we have AI that's going to come in. We have automation, which is always, and new methodologies that are always changing, and then legislation. The government's constantly changing the regulations. We have a dermatology person, and they've changed the way that they're interpreting who can do the tissues and make the slides. And that's actually going to open the door for more medical scientists to do tissues, the education. So realize that this is a changing situation, and we'll just roll with punches as they go. But it's only brighter and more, a greater need, if that makes any sense. Okay, so, do you mind if we spend the last 5 minutes on a case study? Okay, so I'm going to do a case study. So this patient was born with, and I'm gonna go full nerd on you, okay? Buckle up. So this patient was born with a mid-sagittal defect in the body, okay? And so they ended up with a heart condition called pulmonary atresia, which means that the pathway, you guys haven't covered cardiology yet, but the pathway that the blood was pumping couldn't go to her lungs. So at 5 days old, she had to have a bypass conduit put into her body so that she could actually circulate her blood properly, okay? Now, as she grew up, though, what happened was that her blood volume changed, right? And the size of that conduit needed to be bigger for her to be able to deal with that. So what they ended up doing is they went in and do open heart surgery and put a new upgraded conduit into this patient. Well, what happened is they came out of surgery, and obviously the central pallor in their skin wasn't very good, you know what I mean? She doesn't look flush. Well, later that night, the nurse switched, and they had a chest tube to kind of help with just keeping negative pressure, and the nurse reamed that chest tube, and a big clot of blood came out, and then a lot more blood, and then even more blood. So the doctor ordered a couple of units of blood for the blood bank to be able to match with that. Luckily, like the blood kind of like stopped and stuff like that, the bleeding, and they took the cell saver from the surgery and then ended up putting that back into her body. And that's the team that I'm related with. So looking at her hematocrit, her platelets, her white cell count, looking at matching her blood, and looking at everything. And so her life is taken care of, and she's doing just fine. This is my daughter. So, and she's 7 now. This is another picture of her. So, every tube that I touch, I don't really deal with patients directly, right? But I deal with patient samples, and I treat patient samples as they have therapy. So identification is super important, and making sure that I'm applying all the skill set that I have to analyze that as best as possible. So, thank you so much for your time. Any questions?
Did he cover everything on that for you guys? How about your earnings? Not the earnings.
Oh,
You skipped a
so-
slide.
So it is skipping, like it's hiding some slides. Like there's, all the pictures are missing from this. Oh, there it is.
There we go.
Okay.
Just take a snapshot.
Yeah,
of your
yeah, go ahead and
phone.
take a picture of this. And let me explain this a little bit. So they have a processing school where you can go to Intermountain, and actually they will hire you and train you how to do this stuff, the earnings, okay? And you can work, but you have to work for so many years to actually pay back your education. Then once you take that those 2 classes, you can make more money being a clinical laboratory assistant. And then I got my 2-year degree, and I was able to work for the county with that degree, and then leverage more money for that. Now, you can see that these values, these are actually a little bit low, okay? So right now, I'm an advanced medical laboratory scientist, and I'm currently a technical consultant. So mine is actually, I think up to 51, is 51 something, 60, 51, 60 is where I top out. Whereas if you're like a supervisor, a manager, or a professional, you can earn a little bit more money. Yeah, and I don't know why this screen, my clicker was skipping things. University professors can make about $95,000. I've been asked to go up and teach. If you're a dean, you can easily make 6 figures. Now, because I've worked shift work and all those overtimes and overtime hours and holidays and weekends and all that stuff, you can make easily 6 figures doing overtime. Bye, class.
Thank you.
Thank you.
Thank you.
Thanks.
It's a good to remember.
Well, yeah, pages of the lab, and maybe it's because when we were having issues, it started hiding the slides.
Oh, I don't know.
I'll ask you a question.
Yes, what's up?
Do you have to go through like pre-hospital job or
No. So that's the thing too. So clinical experiences and stuff like that. So we did do phlebotomy on each other and stuff like that, but no spinal taps. And so we didn't have to do any like trauma training or anything like that. Now I am certified for like separate and stuff like that because I actually seek it out
Yeah.
because I
Yeah.
But it is very much in the back doors of everything, behind the scenes.
So I've studied like EMS alcohol since I was like 6, and it's a big requirement for us.
Yes.
To have all that trauma stuff. I've done it already, but I was wondering if I went to like something like that, it would help me in college. Probably
So
not, because I'm required 3 years of it.
yeah, so here's the thing. It will help you get in. Does it make sense?
Yeah.
Required is the word that I was focusing on. It won't be required, but it will help if that's something that you like to do.
Yeah.
And I like the flexibility. Sometimes like patients though don't feel like they're mean, you know what I mean? And so trauma, you know what I mean? If you're like an EMT or like a life flight, trauma person. Also a lot of like the PJs from the military are under that same guidance. That's actually where PAs came out of, is that more trauma-based like triage. Whereas this is like on the back end, where we work with the trauma teams. So I would supply blood for the trauma teams to take on the helicopter to go transfuse as they try to save their life and get them surgery. So good question. Yep. Yeah. Take a look at it.
Oh, geez.
Yeah, it's skipped a bunch of the pictures, so it might be in a book.
I think it's sealed, because there's no pictures of your
Well,
lab.
let's go into the, stop playing it, and let's just see if it's in the background. So.
I'll just pull it over and we can take.
Yeah.
Okay. So that is the one that doesn't have, and this is the one, does this one have it? Huh.
So that's why I would think if we went to the current slide, oh yeah, they're not even in there. It's like they got completely removed.
Is this your number?
No, it's not. But
Because this morning I just typed your medical life sciences, this presentation. And this one, I'm thinking I opened the wrong one.
it's okay. Yeah.
Oh, yeah, I opened the wrong one.
It's okay, because it helps me expedite it. And in fact, what I want to do is probably condense these down, if that makes sense. And then that way, like, have all of those, like, forms on one slide, have all of the departments on one slide, if that makes sense, so that they're all together. Because they don't need to, like, you know what I mean? I can expedite that going forward. So we'll do that next year. It's all right. It's all right.
Shoot. All right, well, yeah, that worked out perfect. Doing that smaller class,
Yeah.
hit stop on this one, then I can